So this is part one of lecture 2, where we're going to focus on the cardiac output and regulation of the cardiac output. Now this lecture breaks down quite naturally into 3 sections. First of all, I'm going to give a more detailed anatomy of the heart and a bit more detail about the different structures within the heart and how that relates to initiation of a heartbeat and conduction of a heartbeat. I'm then going to spend some time in part 2 on the cardiac cycle, and finally talk about the cardiac action potential, our action potentials, and how that relates to the electrocardiogram. And for the electrocardiogram, there will also be an online session for that using LabTutor. So let's go to the anatomy of the heart. So I've already given you the basic anatomy of the heart in lecture 1. You'll know that it has 4 chambers, etc. I just wanted to give a little bit more detail about the nomenclature of the different layers of the heart, because perhaps not in this module, but in later lecture modules and throughout your careers, you might hear some stuff referring to the different layers of the heart. So the heart has 3 layers. The epicardium. Epi generally refers to anything facing outwards in the body, and that's the outward layer of the heart. That tends to be connective tissue, etc. It's almost like a protective enveloping package around the heart. The myocardium, if you like, is what most people refer to the business end of the heart. That's shown in the orange on this diagram. And the myocardium is essentially where you have the cardiac myocytes and the muscle cells within the heart. So that's doing the contractile business. And again, the inner layer, the endocardium, again, endo normally refers to things facing inwards when we're talking about anatomy. That's the inner lining of the chambers. So again, that's connective tissue, but it's worth bearing in mind that endothelial cells also line the chambers of the heart. And again, you could compare that to blood vessels, which have a similar 3 layered structure as well, even though the overall anatomy is rather quite different. So let's look at the heart structure again, and I've already described the reason why we refer to as the right and the left. It's as if we're looking at the right or the left side of the heart, so they appear reversed in these kind of diagrams. This diagram, available in the notes, clearly labels all the major parts. I just want to highlight the 4 chambers, and those chambers are divided by valves, and there's also valves on the arteries leaving those chambers. So if we then look at this diagram, now we're looking from above down into the heart. So note that the left hand side of the heart is shown on the left hand side now, and you can see in this diagram clearly how the left hand side of the heart is much bigger than the right hand side of the heart. This just gives you a picture of the major valves in the heart. So we'll start with the mitral valve, and I'll just highlight the mitral valve. That is separating the left atria from the left ventricle, and you can see there is a bicuspid valve because there's only 2 parts to this valve. The tricuspid valve separates the right atria from the right ventricle, so that's shown there, and this contains the valve consists of 3 parts in contrast. If we then look at the aortic valve, what we mean by the aortic valve is shown in this diagram here. That separates the left ventricle of the heart from the aorta, and the pulmonary valve shows separates the right atria from the pulmonary artery. To give you a little bit more detail about cardiac muscle, and you can also look at Professor Stevens' muscle lecture too, and again also at chapter 8 of Pocock and Richards to help define cardiac muscle away from skeletal muscle, even though it is very similar. So it's striated, intermediate contraction speed, the fiber proteins are all there. It's controlled by calcium binding to troponin, and also gap junctions join muscle fibers. So again, if you can't remember what gap junctions are, go back to my cells and their environment lecture. Okay, so similar to skeletal muscle, striated muscle, but also distinct physiologically and anatomically from skeletal muscle. In terms of initiation of a heartbeat, if you remember back to the previous lecture, I showed you that there was an area in the right atria called the sinoatrial node, and that is the pacemaker area for the whole heart. So if we look in a bit more detail and look across to the cells of the sinoatrial node, these cells are different to the surrounding cardiac muscle on the right atrium, and they generate action potentials in a rhythmic manner, and these action potentials of the pacemaker cells are shown in this picture here, and these action potentials move to the surrounding smooth muscle cells. Now these surrounding smooth muscle cells are connected by lots and lots of gap junctions to each other, and that allows electrical signals to pass through the muscle cell layer very, very quickly. So these action potentials in the sinoatrial node trigger action potentials in the smooth muscle cells, and these spread through the smooth muscle cells rapidly, a bit like a cable, via the gap junctions. So pacemaker cell triggers the muscle cells to contract. If we then move to the conduction of the heartbeat through the sinoatrial node, and I've already shown you this figure before. This is lifted from Pocock and Richards, so this is the full figure that you'll see in Pocock and Richards, which gives you more detail about the action potential, and we'll mention the action potential in more detail in part 3. So again, we have the 4 valves or 4 chambers of the heart: right atria, left atria, right ventricle, left ventricle, sinoatrial node, and we're getting the formation of action potentials which move across the atria as shown by the arrows and the arrows that I'm drawing, and they spread across both atria. So what happens is the electrical signals generated in the sinoatrial node cause action potentials. So this is the action potentials there, go on to cause action potentials in the atria, and they spread across. Now, as I said in lecture 1, there is this fibrous skeleton of connective tissue which insulates the atria from the ventricles. So when an action potential has arrived at the atrioventricular node, it doesn't cross across this fibrous skeleton. Instead, the action potential from across the atria is sensed by the atrioventricular node. That slightly is another pacemaker cell that slightly delays the action potential and then sends it down conduction pathways, including these Purkinje fibers, which penetrate through the muscle layer throughout the heart, and then they trigger coordinated action potential and contraction of the right and left ventricles at the same time. So we can see the atrioventricular node sends pulses down to the Purkinje fibers, the conduction pathways, which then causes an action potential and then contraction of the cardiac muscle in the ventricles. So 2 phases.